Yeah, welcome to our mathematical biology seminar today. Today we have Rodica from University Iowa. So without further ado, let's welcome her.
Yes, thank you. It's a pleasure to be here. Thanks for the invitation. I'll talk today about the project that I finished recently with one of my students that he actually moved at SMU and then now is at Oregon, Paik Melland. And at the beginning, I just want to, give the acknowledgements to him because he's done the most of the work with the numerical simulations, and also to the great group of researchers from the Human Brain Research Lab at the Neurosurgery Department here at the University of Iowa who will help with the collection of data, and especially to Kirill Nursky, who was the collaborator in a previous paper, but definitely to Dr. Howard and Kawasaki, who got the recordings during the surgery implants, and Christopher Kovac, who also helped with some of the pre-processing functions, and other members in the lab who contributed with the ideas and discussion of how to look at the pre-processing of the data. So what is all about? You may be familiar with visual stimuli, which produced by stability. So it's the so-called bistable perception. It's a famous experiment with binocular rivalry, where you show the same stimulus to people, but somehow you force one eye to look at a house and let's say the left eye to a face, and then the people who are looking at the same presentation of the stimulus start to report a switch between percepts. So the switch continues for a second, and it's sort of uncontrolled. So you start to report it and you keep going, but that's not really a way to control it, to maybe for a while to force you to see one percept, but then the other one will pop up. So what we've done here, we look at the different kind of stimulus. So tap a different sensory system and presented an auditory streaming of triplets as a stimulus, which induces a similar type of behavior, but in the auditory system. So in the sense of we put together two tones. Let's say I'm gonna call it A here. I assume you see the mouse, right? Hopefully.
Yes.
So yeah, thank you. Okay, so the frequency, a tone of some particular frequency, and that's, let's say the tone is at 100 millisecond duration. There's a short gap of silence for 50 milliseconds, present the next tone at a different frequency level, again, 100 millisecond duration, a short silence of 50 milliseconds, the tone A once more, and then there's a longer gap of 200 milliseconds. So in total, this is like a 600 millisecond duration triplet. And then we assemble those triplets again and again and again into a sequence. Basically here, our stimulus was 5 minutes long, which corresponds to 500 triplets in this sequence. So the tones are fixed to along the 5-minute presentation to the A and the B tone, but this structure of the stimulus produces a change in a perception. Well, when the difference between the tones is in what we call an ambiguous region, so the difference in semitones, I'll actually show a next picture which explains a little bit better. But what happens is the subjects listening to the stimulus here and report two different percepts. One, we call it one stream or an integration, and it looks like more like a galloping rhythm. And segregation is when they hear the two tones kind of in the background of each other. So I would say, if you want to compare, think about the Morse code when you hear the both A and B tones. So I put here together two examples. One is what I call control, because I should mention, we ran those experiments with subjects which were patients having epilepsy, and they were having implants for the for the epileptic surgery procedure. But in the meantime, they also ran a sort of experiments in the lab. So when you work with ECOG patients, you do not have a lot of time to run your experiment. So basically, we had to focus in more like in a half an hour overall time as a session. And you do need, for this kind of experiment, you do need longer stimuli. So at least, like 5 minutes if you want to see changes in perception, because it does take longer time for the perception to build. So for that reason, we assembled two different stimuli. One, we call it control, where we actually forced through the change in the tone frequency the subject to hear the two percepts, to kind of force the percepts on them. And the other one is just a simple bistable stimulus, when we do not manipulate at all the two frequencies, the A and the B tone will have a fixed frequency along the 5 minutes presentation, and they just, the subject report the two percepts. So I'll just show the control because I think it's more obvious. We use the control also like a training session, also to explain, and also to have some idea of the response, of the accuracy of the response for the subject and other kind of properties before we run the bistable stimulus. So let me just show the control. It's kind of annoying, but I'll keep it short. So I hope it kind of gave you an idea about what I mean by galloping, and Morse type of, two segregation percept when you hear the two tones kind of in the background. So let's see. I have for the bistable stimulus the same, so I'll just skip it for a moment and just go to the main question that we try to address. So basically, in this kind of experiments, there are two sources of recorded data. One is the behavioral data, the subject reported button presses. So here for the talk, I would like you to think, and whenever you see the red color, think that this is segregation or the two stream kind of percept. That when you see blue, that's integration, the triplet, the galloping percept. So there are, reports of changing between segregation, integration, segregation again, integration, and so on. And the other type of recorded data, which was done in parallel, is ECOG data, intracranial EEG. Basically, the data was collected from several contacts, but we're going to focus in this talk only on contacts from Herschel gyrus, posterior medial Herschel gyrus, which is the equivalent of A1 core auditory cortex in monkey, for example. So I'll show what I show here. There are 4 contacts in the Herschel gyrus, and how the data looks like in a snapshot from out of the 5 minutes. So these are the two, type of percepts. And the question is if we can, somehow see a signature in the ECOG data of the percept itself. Now, oops, I think I went in a different direction. And ideally, what we'd like to see is some features, extract some neural features from the recordings that correlate with perception. And why we're interested in this? Well, first, most of the data, these kind of experiments and the way the data was analyzed before, even, by our group, but also others, the typical approach is, because this is just, a very long trial where you do have changes in perception, but you would like to see some correlation with the triplets. People are doing are just taking the signal and chop it in epochs based on the triplet length. So if I have in this example 600 millisecond duration, again and again and again the triplet during the presentation, I'll chop the data according to the 600 millisecond duration and have a bunch of epochs. Then, people are splitting them. I color them in based on the perception. Say some of them are, I'm going to call those epochs trials, even if, that's just one single actually trial for the 5 minutes. But now I'm going to redefine the trial based on the epochs and color them based on blue and red based on the perception, and then run some statistical analysis. Can be univariate statistics, multivariate statistics, like some classifiers, trying to distinguish between triplets which were associated with integration in blue and triplets which were associated with segregation, which is in red. So we've done this in a previous study, and we saw, and this was like, it's a similar result which were reported, for example, with MEG data and other studies, that there are some windows during the 600 millisecond epoch where we see statistically differences between the average, let's say, local field potential between triplets in the one percept versus triplets in the other. The problem that I saw it here, I'm coming from mathematics and I have a dynamical system type of background. I always felt that this is not sufficient, because basically here, when we talk about bistable perception, we talk about the longer time scale process. So it's a slow process, and that shouldn't take place on the scale of the triplet. It has to be longer. But there's a difficulty of looking at the data, which in this case is definitely non-stationary for the 5 minutes, right, recording. But if there is another way of looking at the data that might actually identify such a slow rhythm, right, that will be what I will call an extracted neural feature, and that will be like ideal if that it's possible in this kind of experiment. So relate this with, switches in perception, which are basically seen mostly in models, because one approach, in analysis of data is, like I said, multivariate or single-variate statistics, univariate statistics, but that needs many trials, so you have to make them somehow, and that's a way to do it with the epochs. Or, on the other hand, the approach is computational, and there are a lot of models for bistable perception, in general, and there are also models for bistable perception for the auditory streaming of triplets. For example, I'll just cite here James Rankin and John Rinsel paper, where they came up with a firing grain model where you have 3 variables associated with the A tone, the B tone, and A and B tone together. And the principles in this modeling is usually you assume some competition. It's an attractor, competing attractor, with ingredients which include noise or adaptation or both. There are other approaches which rely on Bayesian updates or evidence accumulation. But this is one example which actually shows that if you try to connect data with computational and mechanistic principles, you do need to look at this kind of data at the longer time scale than the time scale of a triplet. So I mentioned before that usually when we play with the difference between the tones, you can promote segregation if it's a large difference in semitones between the tones in the triplet. You can bias actually the perceptual integration, but if you're in the ambiguous regime, that's going to be this exchange between integration and segregation and back, which is what we call perceptual biasability. So pretty much the goal here was to try to look at the data from the lens of dynamical systems and see if we can extract more than what is usually done in the literature, and find features that may correlate to perception. And in order to do this, we came up with an algorithm which basically combines some recently developed algorithm in just one, and I'll explain why we needed to combine all three. It's because the peculiarity of the data here. But just to kind of give you the idea of what the algorithms are doing and what was the idea, I'll just start with an example, which is kind of a simple, a known example of a double pendulum, where if you would like to write the dynamical system, right, you have the coordinates x1, x2 for the red dot, for the blue dot, but usually you represent this in terms of angle, theta1 and theta2 for, with the vertical direction and momentums, and you start with some equation. So if you run this dynamical system, you'll end up, having a pretty complex behavior trajectory in the plane. It's definitely complicated, but let's say you or me, or, in this case, the ladybug, it's just able to see only the projection of the red point on the horizontal direction. So this is basically the point I'm looking at here. So if this is the only thing which is observed or reported, basically this is x1 of t, and this is just an oscillation, an up and down change, but it describes an oscillatory process, but is definitely way far from what the real trajectories and behavior of the system is observed in reality. So the question, a similar type of question can be addressed, for the brain. I mean, what happens is, let's say there is the dynamics of the brain during this type of experiment that I described with the bistable perception. And maybe the dynamics of the brain is represented by a dynamical system, many equations with expectant variables involved, obviously. But what we actually see is none maybe of the x of t's dynamics. We see actually something else. These are the recordings from the contacts when you have implants in the brain. So here, for example, the contacts can be on the surface or can be in the depth, but basically the recordings are local field potentials changing over time from different contacts. So these are observations, but they might not be exactly related with the dynamical system itself. So the question is how, is there a way to learn something about the system itself by just looking at those observations? So the big question usually is, let's say, I have a system, a dynamical system in a particular higher dimensional space, which, I know that is there, but I basically don't know the governing equations. So even more, maybe the points, even if, let's say I'm in R3, they actually stay on a lower dimensional manifold. Okay, that's possible, but I don't know that manifold either. So, as an example, think about the Lorentz system. It's a 3D, right? I have three equations describing the dynamics, but if I'm thinking where the dynamic stays, is actually like a two-dimensional attractor. So what happens is, okay, maybe we cannot have access to the original system or the manifold where the points stay, but maybe we know an observation which changes the manifold into a lower dynamical system, in the lower dimension points, that can be useful to draw a conclusion about the dynamics itself. Well, unfortunately, this is unknown either. What we actually have access to, and thinking in terms of the brain, is the LFP, the measurements from those contacts are some measurements, scalar for each contact, and then if you assemble, you get the vector, obviously, of the contacts, which you can get. So these are the measurements. So imagine the LFPs put in the contact is a vector of, at each time point I'll have a vector y, and then obviously I can just put in a matrix. So these are the things that we know. But obviously, we'd like to connect and learn something about the original system by just looking at those observations. The algorithm that we put together here to analyze this type of data was actually including 3 different pieces which were developed independently pretty recently in the literature. One is like it's called the time delay coordinates, then diffusion maps, and then extending dynamic mode decomposition. So I will just comment a little bit on each of them to explain why it was important, each of them. I mean, in theory, each of those pieces in the algorithm may be very successful for, analyzing a type of data. So for example, time delay coordinates, what is doing is just taking history of the data to try to identify a structure of the points, if you think in terms of the manifold I described it. So we don't know the manifold, but we can learn something about the manifold itself. Now, diffusion maps will help reduce the dimensionality of the system. So it's like I said before, have, for example, that manifold projected on a lower dimensional space now. Will help learning something about the G projection itself. And what about the dynamic mode decomposition? Well, dynamic mode decomposition was kind of successful recently in capturing the dynamics of nonlinear dynamics of system by looking at the data, but it does rely on the fact that you do need many observations in order to apply the algorithm, especially in terms of observation versus time points. And this doesn't work for us because an observation for us takes 5 minutes. So it's like the recording is every one at the millisecond time scale. So, you multiply 5 minutes with seconds and milliseconds, so you get many data points in time. But how many recordings we have, I mean, how many observations? Very few. So it's the number of contacts. So in our case, for some of the subjects, we had about 10 maybe contacts from where we got the recording, some of them 6. So it's a low number. So in order to apply dynamic mode decomposition and take the advantage of it, you do need to create more data. And it's good if you can create it somehow, to create database on the data itself. And that's what the first part of the algorithm is doing. So what about the time delay coordinates? So again, the simplest way to think about is in terms of Lorentz attractor, where you have the x1, x2, x3 variables. But let's say the only observation of the system is through x2, which I represented here. Looking at this, you see oscillations, but you don't know exactly how the points are maybe distributed in the 3-dimensional space. But taking a delay, so a shift of the trajectory, let's say in this case, I just pick one delay, and plotting one versus another will actually introduce some knowledge about how the points, the original points of the trajectory are distributed along the, their intrinsic manifold. So that's a way to, use the time delay coordinates to create more data points, but also, in a way that capture the structure of the data. So the basic idea of the time delay coordinates theory is given in the Takens theorem, which said that, well, if you take a trajectory, on a manifold at some particular dimension, and let's say you have observations of those trajectories, by taking a number of delays, the repetitions of the data, what you're doing through those delays, you create actually a much higher dimension for the data, right? Then if the number of delays is sufficiently large, so that's S, which will tell you how many delays you take. So by taking sufficiently large, then these data points will actually yield an embedding of the manifold itself. So, there are some constraints here. In practice, this kind of inequality can be relaxed. You don't need maybe necessarily this. But again, you don't know the dimension of the manifold a priori, right? So it's sort of a trial and error idea of how many delays you can take there. There's in the algorithm by, where is it? Yeah, but Whitney no, not Whitney, I'm sorry. Later on, it's going to be Barry and others there. They actually suggest that the number of delays, is a parameter based on the data that you can adjust, not to have too fast of the decay in the data, because then you don't take enough history, but not to have too much, because then the data, it's still too noisy. So now I would say that in the case of our recordings, now the observations are the sequences of the recordings from the several contexts that we have, the LFP. For each time point, you create this vector, which is corresponding to the number of contacts, and then take a shift, like a history of the signal, and another shift to create this huge matrix, which appends the temporal history, in the hope that that will actually correspond to the idea from Taken's theorem and capture structure of the data. So that's the idea of the first part of the the algorithm, which by itself, like I said, is very successful in many cases and for many examples. Now, this was helpful for us because, again, we didn't have a large number of contacts or a large number of observations compared to the long time scale of the observation itself. So having the history along this long signal actually helped gaining some information about the structure of the signal itself. I would also say that when you apply this algorithm, what happens is you take data, which was high dimensional already for us, in the sense of how many time points we have for each of the measurements. But by doing this, taking the history and creating matrices even larger, you take the data point in a really even higher dimensional space. So that one presumably captured the structure of the data, but it will be helpful if you can actually visualize and understand what's happening there. So that's where the diffusion maps algorithm comes into play. So the whole idea for the diffusion maps algorithm was to actually project the data in a high dimensional space on obviously a lower dimension. And while you're doing this, the overall idea is to take those data points, try to compare them. So I will just give an example. You might be familiar with PCA, like where you take the principal component, and all those are actually eigenvectors corresponding to some eigenvalues for a big matrix. So basically, comparing the data, like in a Euclidean space, with an exponential kernel will actually make points which are closer, look similar, right? Data points at a particular time point, which are similar, closer together, while because of the exponential, but points which look more different to actually, kind of decay fast the distance between them. So this is actually a typical idea for constructing the so-called graph Laplacian, which allows to construct those eigenfunctions corresponding to some eigenvalues, which obviously in this case are many, right? It's the dimension of the matrix itself, which was very high already, but this will allow to actually, based on the order of the eigenvalues, right? The decreasing order of the eigenvalues, which are always real here, and positive, you can approximate, let's say, the points as a linear combination with maybe only a number of them. So again, like in PCA, we'll just, let's say, assume that you look only at the first two components. Well, here you can actually take more, but you don't have to take all of them. So this will allow you to actually reduce the dimension of the data, by just picking a number of components. Or I can call them features, but I'm going to call features actually at the end of the algorithm when we apply the dynamic mode decomposition. So what's the idea for the dynamic mode decomposition? Well, again, this is actually nice because it will relate the data points from the measurements we had with dynamics. So the first two parts, time delay coordinates and diffusion map, I would say, will give you points on the manifold. So where the points will stay, if you think in terms of, let's say, like in dynamics, the easiest way to compare, I'll just say, consider, like a limit cycle where the curve described by the limit cycle is a circle. So the circle is the geometric object. So I'm looking at the points, I see them on the circle. I'll say, okay, that's kind of the geometric structure. I know what the data points say. But if I would like to know the dynamics, how the points move along the circle, clockwise or counterclockwise due to the dynamics itself, well, I need extra information. So that's what the dynamic mode decomposition is doing. So it's assuming you have a dynamical system and you have the flow map, but then you can maybe transform the flow through a transformation of a function into something equivalent, right? An observation with it. So now instead of looking at the original flow, I'm looking at something equivalent with the flow. And this is the way this operator, which is obviously infinite dimensional because it works on functions, can be analyzed as numerically. It was shown about 10 years ago that it can be well approximated by a finite matrix, and that matrix can be actually constructed using the data. So for example, now if I think about the output from the diffusion maps, there are those eigenvectors, psi1, psi2, psi-n that I picked from the decomposition of the matrix in the diffusion map. I can use them to create like a time point, a matrix, and then shift the columns and create another one, and then assume that this is the linear dynamics between them. So now I have what it's called a Koopman operator, and the theory shows that you can construct again the eigenvectors and eigenvalues and eigenfunction, the eigenfunction, eigenvalues, and eigenmodes for this Koopman operator. And the output is again a bunch of eigenfunctions, which are what we're going to call them in this context features for our data. So I know that it sounds overwhelming in terms of theory, but that's actually what we plan to do. So again, why we needed all these three together? Because extending mode decomposition or dynamic mode decomposition is nice if you can apply, but you need more observations compared to the time points, and that's something that we didn't have. So how to create them? We use the first part through the time delay and diffusion map to create them. So basically we use the original data to find a library, I would say, of functions, those eigenvectors, that we use them to generate actually as an input to the dynamic mode decomposition. Okay, so how are we doing now? A lot of theory here, and that's kind of the idea of the algorithm itself. But now I'll go back to our data, which is putting together single trial data, 5 minutes long, recordings from a number of contacts in the Herschel gyrus, posterior medial Herschel gyrus. That's the core auditory cortex, and we apply those algorithms to 4, 6, 10 contacts the way we had it there. So what we notice, so the output of the algorithm, again, it's going to be the set of Koopman eigenvalues, eigenmodes, and the eigenfunctions. Eigenfunctions in this case will be what we consider the neural features extracted from the data. Also, the eigenvalues can be considered features, and we can look at them. And the eigenmodes will actually tell you more about the spatial distribution of the values themselves, how to reconstruct particular contact, the data from a particular contact in the end. But what we notice is, when you look first at the eigenvalues, that's what we found very interesting, was that they seem to be organized in a particular way. So, all the eigenvalues are complex, so you can take the real part and the imaginary part. Now, for the imaginary part, if you want to see the frequency, you can divide by 2 pi. So what we notice is those eigenvalues organized in 2 different branches. So one type of branch, which I color here dark gray, is showing eigenvalues which are at multiples of the main frequency in the repetition of the triplets. So the triplet is repeated every 600 milliseconds, which corresponds to 167, let's say, hertz kind of frequency. And if you take the multiples of this, the harmonics, it looks like this algorithm picks them in a very nice way. And there's a different branch which has a similar property. It's just that the real part of their eigenvalues comes, you know, if you see it again, it comes down along the order in the real part much lower than the original one. So that has more power than on the second branch. And what is also interesting is the second branch where those eigenvalues were organized, we noticed that there is one eigenfunction which has a real eigenvalue, and that actually is the one who picks up the slow rhythm seen in bistable perception. So this is just an example for one of the subjects, and just to compare what other differences are between those two branches. I would say if you look at the main eigenvalue here, the lowest one in terms of frequency, but the highest in terms of power, right? The real part. On the first branch, there's no difference, no significant difference, no observation between what's happening in between one percept versus another. So look at the blue versus red. So they really look like sine and cosines at that particular frequency, with no distinction between those data points. But if you look at the corresponding eigenfunctions, the features across the second branch, you see modulation when there is a change from one percept to another, and this is consistent in control, in bistable, and actually, so this is for one subject. I'll skip it and I'll show you that this is consistent also for a number of subjects that we look at. So basically, we look at 3 different data from 3 different subjects, each studied separately, and we saw the same specific property in the way the eigenvalues were distributed, formed the 2 branches, and the fact that there was this feature eigenfunction which had this property which correlate with behavior. Now, this actually shows the dynamics of this feature, the eigenfunction. If I'm looking along the time, it's a snapshot from the 5 minutes. Looking at the feature where you see this slow dynamics in the special eigenfunction and changes with behavior. So we try to look at those features and see if it's possible to use them to make some prediction for the button presses, because that would be an interesting way to use the feature, not only to say that there were changes in terms of the amplitude of those features which correlate with behavior, but maybe say something about the shorter time scale of the button press. So capture different time scale of the perceptual bistability, the switches between percept, the button presses themselves. So what we noticed by looking at the data was that actually a more interesting way to look is, yes, these slow features look like they capture this kind of slow rhythm, but it would be nice if you can see a much better separation between the 2 percepts somehow. So what we've done, we look actually at the phase, the relative phase to the rhythm for the first eigenfunction, which actually correspond to complex eigenvalues. So I would say, on the second branch, ignore the green point, but look at the first one, which corresponds to the oscillation. So looking at this phase, we actually realize that can be used as in a predictor to give an idea about where those button presses occur. And the way we've done it, we took this relative phase and we averaged it across 1 triplet and across 2 triplets, because the idea would be that the change when, a change in perception occurs is something happens at the transition from one triplet to another, and define a predictor which actually give a pretty reasonable idea of where the button presses occur. So that's what we showed in this direction. On the other hand, we obviously we wanted to look at the geometric and dynamics description of the data in projection on those features. So what we've done, I should say at this moment that all those construction of the features that I mentioned through the algorithm, through the time delay coordinates, diffusion maps, extending the dynamic mode decomposition, all of those did not take into account the behavioral data. So it's just the algorithm runs blindly to the reports from the subject. You get those features, you can use them. For example, that's what we've done. We use the special feature at the beginning of the second branch, and then the next two, which correspond to the lower frequency repetition, to actually construct representing in 3D and see what is the structure. And after we get the structure to color based on the subject reports. And we noticed that it's a common structure of the way the data was distributed in the dynamics. And this is what we call the double cone, where points which corresponded to integration, which are in blue, are just separated from the points which are corresponding to segregation as reported by the subject, which are here colored in red. So these are the examples of the projection on the lower dimensional space and the features extracted from the algorithm for different subjects, and I show them, for all 6 subjects. And also in the case of bistability, we had 3 more blocks in the experiment, and they still, maintain the structure. And obviously the idea is, if we look at in those feature, which was like a decomposition of the data, how, if I can say anything about the representation of the data in the original plotting of the context. So what we've done, we reconstructed the 3 of the channels that we put in the algorithm based on the linear combination of the features from the algorithm, and plot them now in terms of the coordinates using the channels. And you can see that the structure with the separation in the 2 parts of the manifolds corresponding to different type of percepts, it's still obvious, but on the same time, it's much harder to identify it through just a regular classifier, which will probably look for a separation in hyperspace or hyperplane. And the structure is much more intricate in the case of this type of data. So chances are much lower if you just use the regular type of approach to look at the data to identify differences. Okay, so I think I have a couple of minutes just to kind of give an idea again, to connect this with dynamics of how we interpret our results. So you're probably familiar with normal form for hot bifurcation, which will talk about will be a way to identify limit cycles in a system. So I just took here as a simple example. When I have the parameter, I just put in R squared just to make it positive here. Based on the parameter itself, if obviously the real part is positive, I'm gonna get the limit cycle. If I go into the negative part for the for the eigenvalue, which here is R squared plus I omega, there's no limit cycle in that case. And you can represent the limit cycle in terms of polar coordinates, where obviously the frequency, if I think in terms of the frequency for our cycle, that's going to be like the omega in the normal form. So that's a perfect cycle, right? I mean, limit cycle that will show the oscillation, and that can occur either for the red part or for the blue part. That would be, imagine it's like a section. If you make a cut in this double cone, either on the blue or on the red, you will see this limit cycle which corresponds mostly to the frequency of the input of the stimulus. So, and there's no big difference between the signal itself during the 2%. So what we say in that case is the first branch that we identify through the algorithm is actually picking up the signal, the characteristics of the signal. On the other hand, if you think in terms of what competition models predict is we'll have, let's say, 2 units with a slow process, let's say, between them that will force one unit to be on when the other one is off, and then the switch, and that continues. So if I take, for example, as the difference between those variables, and then I'm going to have a plus and a minus, right? There's a switch between the signal, and that will correspond with to the slow eigenfunction features that we got from the algorithm. And what we say is, well, that shows up on the second branch identified by the algorithm. All the other eigenfunctions on the second branches are actually a combination of those two. So the first branch together with the special vector on the second branch. But how is this possible? It's actually not through addition, it's actually through multiplication. So it's like a modulation of both the amplitude and the phase of the original signal done through the internal dynamics. So just to kind of, give you an idea of how those are actually, created in terms of dynamics. I have here, small video. Let's see, can I make it a little bit? Oops, where's the Zoom? Oh, okay. What did I do? Hello? I'm still here?
Yes, we have. Yeah. We can hear him.
I don't know what I'm seeing here.
We just see a
definitely something else that I'm supposed to. Okay, let me stop sharing. Share again. I'm almost done. Happens. I can see. Yeah, okay. So I guess I was somewhere here. Yes. So, how the trajectory will move, I guess that video shows, and how you create the, moving from one part to another. So basically, just to summarize, we apply those data-driven techniques which combine 3 types of algorithms into one due to the structure of the data itself to look for dynamic features in the data and dynamics in the data and we derive those features during an experiment which characterized by stability in auditory perception. And I guess the overall conclusion in the end, by just looking at the type of structures where the data sits in when you compare different channels, different contexts in the data from different contexts recording shows that the intricacy of the data points to a complicated process that cannot maybe be achieved in an easier way with simpler classifiers. So we need to take into account algorithms that actually rely on the data itself and the dynamics of the data to capture significant features which are representative. Thank you. I think that's it. Yeah. Hello?
Yeah, thank you.
It's an echo. It's just for me.
For everyone, I think.
So,
all right. Well, thank you very much for this very interesting talk. Can you hear me, Rodica? Yeah?
Yes.
The echo's gone. Okay. That was really interesting. So I don't know, we can take questions from the audience and also the participants the attendees that we don't see, but they can. Okay, so Yangyang, yeah. I don't think we can hear you. No, we can. Yeah.
No, I guess it says Zoom with. Is it true?
There's a bad echo.
Hello?
Yeah.
Zoom said, oops, echo.
All right.
I guess I'm still.
Rodica.
Yes. Is it something with me? Because I don't think I have anything here.
I mean, let me. Can it be me? I'm the only one with the sound on, but I don't hear my echo.
Maybe we'd have to listen. Oh.
All right. Well, I guess so.
Somehow my computer just got locked in something.
All right. Well, in any case, I don't know if there is any more questions. That was really interesting, I guess, to find these structures and connect with dynamical systems. Really enjoyed the talk. I don't know if anyone has other comments, and then I'm gonna read Yangyang's question. So,
Okay.
So a clarified question: is the predictor you mentioned using only the eigenvector associated with the second eigenvalue along the second branch?
Yes. We can get the predictor by picking other eigenvectors, but the one which corresponds to the highest power of second branch is the one who helps define the predictor the best. And if I can, without the echo, mention something. I've seen there's a recent paper on PNAS by Peter Thomas, and we talked recently about this. They actually look at eigenvectors and eigenfunctions of an operator, the forward and backward Kolmogorov operator. My understanding is that the backward operator is similar with the Friedman operator, and they actually show theoretically that this eigenfunction, which corresponds to the eigenvalue with the highest real part on the branch, is the one who capture a lot about the stochasticity of an oscillator in noisy data. So it seems that what we got from the algorithm is similar with what they predicted through a theoretical approach. And we're hoping to understand a little bit whether our predictor is actually related to what they got theoretically. Hello? Do you hear me?
I think now, do we still hear echo?
Hello?
Yes, we hear you.
Oh, okay.
Yeah, thank you. I think now finally the echo is gone for some reason. Yeah, we can take more questions if we have any. I think we have 1 more minute. Let me check the attendees.
Can you hear me now, or you can still? Can you
Yeah,
hear me?
I can.
Okay.
Oh, I
Just
see.
another quick question. Directly try to apply extended DMD to the data, because you have long time series, so maybe your data is big enough.
So DMD,
Yeah, the last one.
yeah. So we tried. The problem is, the way I see it is, so DMD usually, in order to capture dynamics in data, they have this constraint, or it's not constraint, it's like a hypothesis, right? That they use for the data, is that you have the number of observations much larger than actually, I mean, the observation in the sense of context, right? So let's say the recordings, number of recordings from different sites, how many sites, spatial sites, how many I have, larger than actually the observation with respect to time. In our case, was the opposite. We have really long time observation, and I didn't have enough recordings because I had a really low number of contacts. So I would say, I think the DMD can work, especially the extended DMD, the extending dynamic model decomposition, can work probably reasonable well if you do have a higher number of recordings as trials that you can put in the data. We just didn't have, because the econ data, we didn't have access to many recordings. But let's say you have measurements of local field potential from 100 neurons. That's enough number, right, to maybe run your DMD without actually needing to construct, use the first part of the algorithm. We use the first 2 parts in the algorithms to actually feed into the DMD enough observations. So that was the reason, actually. But again, that was part because of we were forced due to, how much data we had access to. But, yeah. So there's no echo now.
Yeah, good.
That's good.
We have more questions. So if not, let's thank Rodica again.
Yep. Thanks.
Thank you.
Thank you, Rodica.
Thank you.
Bye-bye. Okay, it's not here.
Thank you.